Hello everyone. Pharmacogenetics is the future of healthcare. But let's be honest, the amount of data is overwhelming. How do you separate a well supported clinical recommendation from a research only finding? In this video, I'm sharing my curated list of pharmacogenomics resources that will save you hours of search and help you provide safer and more effective care. Today, we are going to talk about a huge conflict brewing in medicine. It's all about this question: what kind of evidence is really good enough when we want to use our personal genetics to pick the right treatment? Okay, think about it like this. It's pretty routine for a doctor to adjust your drug dose based on your kidney or liver function, right? They see a lab result, it makes biological sense, and they make a change simple. But when genetics gets involved, we suddenly throw up this massive wall and demand an almost impossibly high level of proof. It's a real double standard, and that creates this paradox, which brings us to the big question for today. How is that the supposed gold standard for evidence, the randomized controlled trial, is actually becoming a major roadblock for personalized medicine? I mean, isn't that the whole point of this field? Alright, let's break down this bottleneck. The whole problem really kicks off with some simple but pretty mind-blowing math. Yeah, just look at that number, over 900,000. That's how many people you would need in a classic clinical trial just to prove a modest benefit for one single genetic variant. Can you imagine? For most conditions, a study that massive isn't just crazy expensive, it's basically impossible to pull off. And look, it's not just about the crazy number, there are other huge problems here for one. These giant trials often use these perfect ideal patients that don't look anything like the complex people doctors see every single day. But even bigger than that is that ethical dilemma. I mean, really think about this. If you have solid biologic reason to believe it, gene could cause a terrible, maybe even fatal drug reaction, is it even ethical to put someone in a control group and just ignore that risk? It's a tough question. So, all this disagreement over what good evidence even is, well, it creates a ton of friction, and it's happening between the exact people who are all supposed to be on the same team trying to make things better for patients, and you can see why when you look at who's involved. Everyone's got a totally different priority. The big guideline committees, they are laser focused on those gold standard trials. The people actually trying to implement this stuff in hospitals, they are just trying to deal with the day-to-day reality. And then you have got the clinicians who are focused on the one patient sitting right in front of them. And of course, the patients themselves just want to know they are safe. Everybody's pulling in different direction, and it's no wonder it's so hard to get coordinated. Okay, so if the classic RCTs, the bottleneck, what's the way around it? Well, the solution isn't to throw out the idea of evidence. Instead, it's about building a smarter, more systematic way to look at all the evidence. We have not just clinging to one single type, and this solution really comes in pragmatic evidences and observational trials. And this solution really comes in two parts that work hand in hand. First, you have got Pharmacogenomics. It's the massive knowledge base that explains why a certain gene affects a drug. Then you have CPIC. There you can see how team takes all the knowledge and turn it into practical guidelines that tell doctors how to actually use it. So let's start with that foundation Pharmacogenomics. This is the pharmacogenomic knowledge base. It's been around since 2000s, based at Stanford, and its one job is to do the heavy lifting, collecting, curating, and just making sense of this mountain of data on how our genes and drugs interact. Then you've got its partner, CPIC, the Clinical Pharmacogenetics Implementation Consortium. This is where the rubber meets the road. It's a huge international group that takes all that amazing knowledge from PharmGKB and translates it into clear evidence-based guidelines that doctors can actually use. And we are not talking about a small committee here. This is a massive operation with hundreds of members all over the world, all working to make this science practical. And they are absolutely making a dent. Just look at the progress. We are talking dozens of official guidelines covering 34 different genes and over 160 drugs. They are systematically turning this incredibly complex genetic science into straightforward advice that can be used in the clinic. So that raises a pretty important question, right? How does a group like CPIC decide when there's enough evidence to actually tell doctors to change how they prescribe a drug? Well, they don't just wing it. They have built a very clear, very transparent framework to make these calls. This is their framework. Basically, every gene drug combination gets a letter grade. It's pretty simple. Levels A and B are the green lights. The evidence is strong enough to do something for A. It's a should. The genetic info should be used for B. It's a could. It's recommended. Then you have C and D, which are basically the hold on. Let's wait for more data categories. It's a super practical roadmap, but they don't stop there. It actually gets even more detailed because within those A and B levels, they add another layer of nuance. They grade the actual strengths of the evidence for every single recommendation. And this is how they do it. A strong grade means the evidence top notch and the benefits clearly went out. Moderate or optional means, hey, the evidence is pretty good, but it's a closer call on the balance of benefits vs risks. So there's more room for a doctor to use their own judgment. It's all about providing that extra layer of context. Okay, so this whole system is really impressive. Every CPIC guideline follows a standardized, rigorous format to ensure clinicians have find information quickly. When you open a guideline, you will find introduction and literature review, gene background, how genetic variations translate into specific drug-related phenotypes, and therapeutic recommendation about clear dosage adjustments and clinical concentration, risk assessments, potential benefits, risks or therapy modification, and other important links. Let's look at how we assign phenotypes using CYP2C9 as an example. We use an activity score system to categorize patients. Normal metabolizers typically carry two normal function alleles, star 1, star 1, with an activity score of 2. Intermediate metabolizers, IM, these patients carry one normal and one decreased function allele, like star 1, star 2, or star 3. Their activity score ranges from 1 to 1.5. And there are poor metabolizers who are carrying one no function alleles plus one decreased function allele or two no function alleles, like star 2, star 3, or star 3, star 3. Also, CPIC uses a flowchart to prioritize testing and clinical action based on the level of evidence. For level A and B, evidence is strong. Prescribing actions or alternative therapies are highly likely to be effective and safe. For level C and D, for level C, evidence is weaker or the prescribing change isn't clearly defined. Testing is common, but action is not always mandatory. For level D, testing is rare and there is no currently recommended prescribing action. If a drug gene pair isn't yet in CPIC guidelines, we look to external groups like the FDA or Pharmacogenomics or professional societies to determine if the pair is actionable. To see this in practice, let's look at paroxetine and CYP2D6 dosing logic. For ultra-rapid metabolizers, increased metabolism leads to low plasma concentrations. CPIC recommends selecting an alternative drug not metabolized by CYP2D6, but this recommendation is moderate level. The same type of recommendation when a patient is poor metabolizer. Then CPIC recommends reducing the starting dose by 50% and maintaining a slower titration schedule. It's worth to mention, CPIC doesn't work in a vacuum. The Dutch Pharmacogenetics Working Group, DPWG, established in 2005, provides a massive complementary ecosystem of recommendations. So the timeline of these resources shows incredible growth. In 2000, Pharmacogenomics launches. 2010, CPIC is founded. 2015, introduction of tools like PharmCat and PharmVar. Finally, in 2025, the launch of ClinBGX, a comprehensive hub that integrates Pharmacogenomics, CPIC, PharmCat, and PharmDoc into a single unified platform. The CPIC and Pharmacogenomics have totally different jobs, but they are working towards the exact same goal, making sure prescriptions are safer and more effective. So, for the longest time, you kind of had to jump back and forth between these two powerful resources on your own. But now, thankfully, a modern solution brings them both together. So let's meet your new navigator. And here it is, the ClinBGX. This is the tool that acts as your clinical dashboard. Just think of it as the navigator. It pulls the actionable guidelines from CPIC and all the deep evidence from Pharmacogenomics into one single clinical interface. That's actually designed to be used at the point of care. Super important. It's not replacing them, it's just integrating them. Now, if you are a long-time user, you have probably gone to the Pharmacogenomics homepage recently and noticed it redirects you here to ClinBGX. First thing, don't worry. All that amazing, powerful data you are relying on is absolutely still there. It's just being recognized and streamlined to make it way easier to access in a clinical setting. This update also came with a few name changes. In our next video, we will navigate ClinBGX platform to see exactly how you use these tools to get a clear answer fast. So, subscribe to be with us. So finally, let's talk about FDA resource. We have more than 250 drugs features pharmacogenomic information, and they are official labeling. The four pillars of pharmacogenomics in clinical utility: drug exposure, managing clinical response variability, and adverse events, mitigating severe risk factors, and precision dosing, implementing genotype specific dosing guidelines, and finally mechanisms to clarify mechanisms of drug action through polymorphic, target, and disposition genes. Also, pharmacogenomics recommendations are heavily concentrated in oncology and psychiatry. About 47% of all pharmacogenomic labeling is concentrated in these two therapeutic areas. In 2020, FDA released this tool, which divides medications and gene association into these three distinct sections based on data support. So in the base, we can see that labels which demonstrate a potential impact on pharmacokinetics properties only, and on the top we can see labels which support therapeutic management recommendations. So, from Pharmacogenomics to CPIC guidelines, these resources help us turn data into better decisions. If you found this helpful, hit subscribe and join me for more insights into the science shaping tomorrow's pharmacy. Precision medicine starts here. at Next Gen Pharmacists. Don't miss the journey. Goodbye.